Hello everyone, welcome to this lecture on the Golgi apparatus. In this session, we will cover everything you need to know about this essential organelle, from its structure and functions to transport mechanisms, associated diseases, and differences between plant and animal cells. By the end of this lesson, you will have a complete understanding of how the Golgi apparatus or Golgi complex operates within the eukaryotic cells. So let's begin. The Golgi apparatus, also known as Golgi complex or Golgi body, is a membrane-bound organelle that exists in all eukaryotic cells, meaning cells that have a nucleus and other specialized compartments. Its primary function is to modify, sort, and package proteins and lipids that are produced in the endoplasmic reticulum. Once modified, these molecules are either sent out of the cell or directed to specific organelles for use within the cell. In simple terms, the Golgi act as the post office of the cell, processing, distributing, and the cellular packages. The Golgi apparatus was first discovered in 1898 by Italian scientist Camillo Golgi, who observed it in the nerve cells. Under the microscope, it appears as a stack of flattened membrane-bound sacs called cisternae. You can see in the image. One of the most unique features of the Golgi is its polarity, meaning it has a functional directional structure. So the cis phase or cis Golgi network is located closest to the endoplasmic reticulum and acts as the receiving side. The medial Golgi is in the middle where proteins undergo processing. Finally, the trans phase or the trans Golgi network is oriented toward the plasma membrane and is responsible for sorting, shipping, and shipping the processed products to their destinations. Let's now look at the structure in more detail. The Golgi apparatus is made up of three main parts. First, the cisternae, which are the stack-like flattened sacs where the processing happens. In animal cells, there are typically 5 to 8 cisternae per stack. Second, we have vesicles, which are small round membrane-bound transport containers that carry proteins and lipids between compartments. And the next one is lumen, which is the internal space inside each cisternae where the chemical modifications take place. This organized structure supports the Golgi's ability to perform complex tasks efficiently. The Golgi apparatus operates through a functionally organized layout divided into regions. So the cis Golgi is entry point and it receives transport vesicles from the endoplasmic reticulum through an intermediate area known as Golgi intermediate compartment or ERGIC. Once inside, the proteins move to the medial Golgi where the major chemical modifications occur. Finally, they reach the trans Golgi, where they are sorted, packed into vesicles based on their destination. This polarity, cis to medial to trans, ensures the step by step processing of cellular materials. The Golgi apparatus performs several essential functions. First is protein modification. For instance, it adds carbohydrate groups to proteins in a process called glycosylation, forming glycoproteins. It also adds sulfate groups through sulfation, which is an important in molecules like proteoglycans. Phosphorylation, the addition of the phosphate groups, is another modification that affects protein activity. Second, it sorts, packages proteins destined to be secreted followed the secretory pathway either while others are like digestive enzymes are tagged with mannose 6 phosphate for delivery to the lysosomes. The Golgi also process lipids such as synthesizing sphingomyelin, and in male gametes it plays a critical role in forming the acrosome, a cap-like structure essential for the fertilization. Transport to and from the Golgi apparatus occurs through vesicles. When materials move from the endoplasmic reticulum to the Golgi and then to the plasma membrane. This is called enterograde transport. In this case, vesicles coated with COP2 protein carry the cargo from the ear to the Golgi. From the trans-Golgi, clathrin-coated vesicles carry materials to endosomes and lysosomes. In the opposite direction, we have retrograde transport, where COP1 coated vesicle bring materials back from the Golgi to the ear, especially proteins that were mistakenly transported forward or that need more processing. So this dynamic movement ensures quality control and correct delivery inside the cell. Although the Golgi apparatus is present in both plant cell and animal cells, there are some differences. In animal cells, usually a single Golgi stack is found near the nucleus and close to the endoplasmic reticulum. In contrast, plant cells have multiple Golgi stacks known as dictyosomes that are spread throughout the cytoplasm, while the core function, modification, sorting, and packaging remains the same. The plant Golgi has an added role. It is also involved in the synthesis of cell wall polysaccharides such as pectin, hemicellulose, which are essential for building and maintaining the plant cell wall. Malfunction of the Golgi apparatus can lead to serious diseases. For example, eye cell disease or mucolipidosis type 2. So it occurs when the Golgi fails to add mannose 6 phosphate to lysosomal enzymes. As a result, these enzymes do not reach the lysosome, instead are secreted outside the cell, causing cellular waste to accumulate. Also, in neurodegenerative diseases like Alzheimer's or Parkinson's, misfolded proteins can disrupt Golgi structure and function, contributing to cellular stress, nerve cell damage. So, these examples highlight just how vital a properly functioning Golgi apparatus is for the cellular health. Let's quickly summarize what we have learned. The Golgi apparatus modifies, sorts, and packages proteins and lipids for transport. It consists of stacked cisternae and operates with distinct regions, the cis medial transphases. Transport between compartments is managed by vesicle coated with COP2 and COP1 or clathrin depending on the direction and the destination. In plant cells, the Golgi apparatus appears in multiple stacks and play a role in cell wall formation. When the Golgi does not function properly, it can lead to metabolic disorders and a neurodegenerative disease. So if you found this video helpful or it helped you to understand about the Golgi apparatus in a clear and organized way, don't forget to give it a thumbs up. Hit that like button and subscribe button to the channel for this channel, and to get more high yield lectures on cell biology, physiology, or medicine or pharmacology, this kind of subjects. Keep exploring, keep questioning, and stay tuned for the next video. Thanks for watching.